So hello guys and welcome back to Excel Academy. So this is our last topic and we were just discussing about the telomerase. So we just discussed like what the telomerase are. It's a ribonucleoprotein. It's a species dependent telomere repeat sequence. It is depending on that. And then we have also seen the differences in the sequences, like very slight differences there, but overall the sequence is conserved. Okay, so we were talking about the human telomerase structure, right? So if you see in the structure, you can see that you have got over here the TERT. So you can see that this is the biggest domain which is present in the entire telomerase, right? So what is this? This TERT is your telomerase reverse transcriptase. Okay, it is telomerase reverse transcriptase. Then you have got telomerase RNA or TERT. Where is it? Here, this one. That is your RNA, right? So you can see that the main is your this particular domain, which is your telomerase reverse transcriptase, and to that we have also the RNA. Okay. And then later we have got the other subunits like discarine is there, GAR1 is there, reptin, pontin, NHP2, NOP10, PCB1, all these other different subunits are present. Main is your TERT and your TR. Okay. Now, it's a reverse transcriptase, which is class of enzymes that create single-stranded DNA using single-stranded RNA as the template. So that's why in the previous lecture, we have said that the telomerase is also a reverse transcriptase. Okay. Okay. Now talking about the sheltrin. So sheltrin is really very important. Okay. And it is like, if possible, try to remember the components of the sheltrin. If not the function, but at least the components. So it is also known as telosome. It is also known as a telosome. It's a protein complex which is known to protect the telomeres in many eukaryotes from the DNA repair mechanism. Why? Because otherwise it will think that it is double strand break. Okay, and it might undergo DNA repair. So to prevent from there, we need this sheltrin complex also. It also regulates telomerase activity. That is, it helps in recruiting the telomerase in the stem cells. Okay, so the absence of sheltrin causes telomere uncapping and thereby activates damage signaling pathways, which is like non-homologous end joining, homology directed repair, end-to-end fusions, genetic instability, senescence, or apoptosis. So few of these DNA repair we'll discuss in the upcoming lectures. Okay. Just for now, just try to remember that sheltrin is important in capping the telomeres, that is to protect from the DNA repair mechanism. Okay, so now talking about its units, subunits, the sheltrin subunits. So in the previous figure, let me just have a little bit idea about this. That is like it has got TRF1, 2. Okay. So if you see that TRF1 and 2 are associated with the sequences over here. Can you see it on the telomeres? They are associated with the sequences. Which one? TRF1 and 2. And these two are associated with the TIN2. Okay. Then we have got the TPP1, RAP1 and POT1. Now, keeping this in the mind, so we have got these different subunits. Just now we have just mentioned. So we have got TRF1 and 2, POT1, RAP1, TIN2, and TPP1. So first, let's talk about TRF1 in detail because this is the main subunit, TRF1 and TRF2. Okay. So if you could remember about the TRF1, then it is easy to remember about TRF2 because they are all almost similar. Okay. So TRF1 means what? It is a telomere repeating binding factor 1. Okay, please remember the full form. Telomere repeat binding factor 1. It's a homodimeric protein which binds to the double-stranded TTA triple G region of the telomere. Can you see? It is binding to the double-stranded region. Okay, it's not binding at the end of the telomere where it is single-stranded. It is binding at the where it is double-stranded. Okay. Now, along with TRF2, it normally prevents the telomerase from adding more telomere units to the telomere. So it means what? TRF1 and 2 is preventing the telomerase activity. Okay, is neither what will happen otherwise? So Otherwise, the length of the telomeres is going to be more in lengthy. Okay. Guys, just stop me in between if suppose any points you're not able to understand. Okay. So now, but when the telomere lengthening is required, okay, that time TRF1 is going to recruit the helicases and interact with transkinases to facilitate the process. Now, this is also important point. Okay. So if you want the, for example, in the case of cancer, ALT is alternate lengthening of the telomeres. Okay. So suppose this is overexpressed, what will happen then? The TRF1 is going to recruit the helicases and the transkinases to facilitate the process. That is, telomerase activity आपका enhance हो जाएगा. Okay. Now TRF1 is highly expressed in stem cells. It is essential for generation of induced pluripotent stem cells. So we know, okay, it is required. Now it is upregulated in the cancer cells in humans and mice because of stem cell quality of the cancer. Okay. Now why do we say that it has got the stem cell quality of the cancer? Because it is also stem cells kaise hain? They are also growing. Okay. The only difference between the stem cell and cancer is that stem cell may be आपका बार-बार grow होता है, repetition, proliferation is there, but it is in the controlled manner. Whereas in the case of cancer, the proliferation is in the uncontrolled manner. Okay, this is the only small difference which makes a huge changes. Now let's coming to the TRF2. So what is TRF2? Again, telomere repeat binding factor 2. So this is going to be similarly, structurally similar to your TRF1. So this is your TRF1, this is your TRF2. So if you see, in both the cases, it is the homodimer. Okay. It is a homodimer. Now, so now it is along with the TRF1, it helps to form the T loops. Okay. Now it is a homodimeric protein. Again, it is binding to the double-stranded region of your telomere. Now, overexpression of TRF2 leads to telomere shortening. Okay. And loss of this leads to the T loop, which is going to activate your P53 and 8A mediated apoptosis. So this comes later when we'll talk about the regulation, जैसे भी हम DNA repair में पढ़ेंगे. Okay. So for timing, just remember that these TRF1 and TRF2, they go and bind to the double-stranded telomere structure, okay? And they are responsible for forming the T loops. Now, what is T loop? So if you see the telomeres, they actually form a loop-like structure with the help of the sheltrin. जैसे यहाँ पे आप देखोगे, एक तरह का structure वो form कर रहे हैं, ठीक है? So this is how, along with the sequence and these proteins, they actually are forming a cap-like structure, okay? A capping structure is formed, which is preventing from the damage. इतना clear है guys till TRF1 and TRF2?
Yes, ma'am.
Okay. Now then we have got the RAP1. It is a repressor or activator protein. It is a stabilizing protein. Okay, so you can see that it's a stabilizing protein of TRF2. Okay. Now one more thing is that, of course, if it is repressed or activated, depending on the situation, it is going to respond. कि cancer बनना है या stem cell में होना है. So depending on the situation, it will be repressed or activated. Then we have got the POT1. So here you can see the POT1 is there. Okay, POT1 is for the protection of telomere 1. So it contains OB domain. Okay, what is OB fold? It is the oligonucleotide oligosaccharide binding. So this will bind POT1 to a single-stranded DNA. So we till now what we have saw was TRF1 and TRF2 were binding to the double-stranded telomere. Okay, only the POT1 is associated with the single-stranded DNA region of the telomeres. Okay, and it is helpful in the formation of your D loop, that is a displacement loop. Now coming to the TPP1, it is associated with your POT1. So when telomeres are to be length, यानी कि उनका जब length हमें बढ़ाना है, so TPP1 is a central factor in recruiting the telomerase. So TPP1 is required for recruiting the telomerase. Okay. Now last 10-2. So that is TRF1, TRF2 interacting nuclear protein. So it is a stabilizing protein which goes and binds to both your TRF1 and TRF2 and helps in making a complex with the POT1. Okay. इतना clear है, structure clear है अभी आप सबको, the sheltrin complex. Yeah. So we have discussed about the TERT, we have discussed about the TR, that is telomerase reverse transcriptase हो गया, ये आपका RNA template हो गया यहाँ पे, ठीक है? Okay, then coming to the sheltrin. Sheltrin में we have got the TRF1 and TRF2, which goes and associates with the double-stranded telomere. Okay. Now they help in the formation of T loop and T loop. TIN2 क्या है? It is your TRF1 2 interacting nuclear protein. Okay, so they help in stabilizing your TRF1 and TRF2 and TPP1 etc. Then coming to the POT1. POT1 क्या करता है? वो आपका single stranded telomere को bind करता है, ठीक है? And now this is responsible for making the D loop, and then TPP1. So you can see the association. It is the association of TRF1, TIN2 and POT1 which is coming, which is brought together, okay? And then TPP1 is also responsible for recruiting your telomerase. Clear guys? सारे सब यूनिट का फंक्शन क्लियर है आपको sheltrin में?
Yes.
Okay, now look at over here. So what do we see here? That you have got the telomere, okay? And this is how the sheltrin protein will come together and they will start binding and they form a T loop structure. Okay? इस तरह से वो fold हो जाएगा. So this entire thing is the capped one. Okay? The capped telomere we say. Okay. Now look at this figure. Take your time and tell me what does the first figure represents and what does the second figure represents. And of course, we are talking about telomeres. So it is associated with the telomeres, this figure. Yes. So what is happening? What does the first figure represent and what does the second figure? Okay. Yes, Akshaya, what do you think? No one? Shruti, Tanvi? क्या लगता है फिगर देख के?
Ma'am, I think that the second figure has the genetic material ligating because they're forming loops and the little dots, they're all joined together. So I feel like there's something wrong with the telomerase in the second picture, but the first one, everything is clear and everything is managing to keep its distance.
Okay, Shruti, I don't know whether you really meant that. First figure is normal chromosomes where the red and
so
the
everything
green
is
are
like,
the So red and green dots of the fluorescence, they show that they are the telomeres. Okay. Now,
yeah, and the second one, everything was ligated together. So I thought that.
it is fused.
Yeah.
My God, the terminology. Okay. This is going to be a very big problem for you, Shruti, in future. Okay. You need to learn all those things. So, I mean, I agree that you give the correct answer, but the word usage is wrong. Okay. You can't say ligated. Okay. So they are fused. So what does it mean over here? The 2-3 chromosomes are together fused. If we see over here, they all are fused. So you can see, that's why you can see a telomeres in between. Okay. So this is having a genetic problem then. This person is having a genetic problem then. Okay. Okay. So now the figure is clear. Okay. So this is how you need to learn, like, you know, कि कुछ भी data अगर आपके सामने in future में आता है. Okay. When you're going to do the research, you should know how to interpret the data. So obviously we were talking about the telomeres. So here the main focus will be the telomeres. And we can see that in the chromosome, हम हमेशा कैसे बनाते हैं? ऐसे ही बनाते हैं ना? तो ये टीलोमेर्स हो गए, यहाँ हो गए, यहाँ हो गए, यहाँ हो गए. Okay. Now यहाँ पे मैं एक और छोटा सा डाउट पूछती हूँ. If you observe each case, okay, chromosome, you can see that यहाँ पे एक रेड कलर है और ग्रीन कलर है. रेड एंड ग्रीन, यहाँ पे ग्रीन एंड रेड है. Can you tell me why the fluorescence is like this? Oppositely ऐसे red red green green, opposite में ऐसे है. Can you tell me the reason?
telomerase acts on only one of the strands, right? So depending on which one has the D loop and which one, as in which one is getting capped and which one is staying the same, probably the reason why the
No, no, Shruti,
orientation
say
is
once again, just say once again.
since telomerase acts on one the lagging strand only, right? So it
Huh.
forms the D loop over the leading strand, and the
Okay.
same, but the opposite happens on the other end of the chromosome. So maybe that is the reason why they are oriented in opposite directions.
Okay, but here we are not discussing like how the telomerase or the sheltrin complex is acting. Okay, it's your choice. It's your choice. So of course, on the chromosome also, ये एक direction का होगा. So obviously उसके opposite में ही आपको दूसरा मिलेगा, right? The thing is, this binding of the fluorescence is totally on you. What kind of fluorophore or chromophore you are taking and using.
Yeah.
है ना? So it depends. I mean, there was no any specific reason or related to telomerase. It's just that what kind of tag you're using. So आप जो भी use करोगे, definitely, for example, if I say that I was using the 5' end वाला something which is having the fluorescence. So अगर ये 5' end यहाँ का मैं use कर रही हूँ, तो उसका opposite में ही आएगा, right? Yeah. So depending on that, अगर suppose मैंने 3' का कुछ use किया है, so उसके opposite में आएगा. Something like that. So the choice of using fluorescence, fluorophore or chromophore is totally on you, on the researcher. Chalo, good. Okay, so we are going good. You guys are getting the point now. Okay. Now let's just see a small, already we have discussed this thing, the role of telomeres in aging. Just see again the introduction. So in the normal cases, we know that the telomerase is absent. Okay, and we know that the cell is going for 50 to 70 cell divisions. And this particular thing is also known as Hayflick limit. Have you heard of this Hayflick limit?
Yes.
Okay, good. So you guys know what is Hayflick limit, right? Okay. So they had like many people were there, like especially if you see in India also nowadays people have started taking Botox. I hope you know all this thing. So
Yes,
they take it on their cheeks and on their forehead, everything, right? So what is happening? They're actually taking the space which has been created because of the wrinkles. Wrinkles क्या है? The loss of cells from that particular region. ठीक है, अगर किसी के चेहरे में अगर wrinkle ऐसे आ रहे हैं, it means what? Those cells are being lost. वहां का कोलाजन चला गया है. वहां पे जो cells होने चाहिए replace करने के लिए old cells को, those are not there. Okay, so that gap is finally looking like a wrinkle. Okay, अब आप botox लेते हो, injection लेते हो, so what is happening? The gap is being ऐसे filled, and that's why the person looks younger. अब wrinkles नहीं दिखेगा तो obviously that person will look younger, right? So especially in South Korea, अभी तो मैंने इंडिया में भी काफी केसेस सुनने लग गई हूँ, and of course outside भी होगा, अमेरिका में भी होगा, जापान में भी होगा. I don't know. I know mainly about the South Korea and yes, India also many cases of Kajol, Shah Rukh Khan. आपने सुना ही होगा that they are taking this so that face lifting, face uplifting or lifting कहते हैं, I don't know, just to remove the wrinkles so that they look younger. And for the Bollywood celebrities, that is the only way they can earn money, right? So nowadays I have seen like many other people like who can afford, and since it doesn't have that much side effect, it's just that later on it is going to be very bad. The thing is that if you take more of Botox, what will happen? It's kind of solid thing which is inserted between your cells. Eventually your flexibility of your muscles will be gone. So what will happen? It will be very difficult for you guys to speak. Achha, I'll give you one example. Have you seen Hema Malini talking? कभी ध्यान देना, okay? I'm not against of anyone. That is their wish. जो use करना है करें. But when you take more of these things, it becomes really difficult for you to move your cheeks, okay? And because of which, what will happen? The difficulty in speaking. So ये सारी चीजें हैं. Okay, now coming to the science part. So there are many people, many researchers who are thinking that If we can give telomerase, okay, we can synthesize telomerase using molecular cloning. Like in the bacteria, you can put the gene. The bacteria can, divide and transcribe and translate this gene into telomerase, and finally we can harvest. Okay, we can do the protein engineering. We can do and we can get it. So हमारे पास अभी trans telomerase बहुत सारा हो गया. So if we could directly inject into the system, into the organism, will it act as an anti-aging therapy? है ना? Are you getting the idea? कि what they have thought as an anti-aging therapy हम कैसे कर सकते हैं? It's better to give the telomerase itself, right? But what is the problem if I give so much of telomerase to the cell in where the telomerase should not be present? वहां पे अगर हम telomerase दे देंगे तो क्या होगा?
Cancer.
Very good. So the chances of having cancer will increase. इसलिए अभी तक we don't have as such the anti-aging therapy. You must have heard that to look good and to look young, always drink lots of water, right? Eat which is healthy, which has got, antioxidants, etc., etc. All this is true because these antioxidants are actually, to keep your enzymes healthy. Okay. Now, if you eat trans food, fried food like in a McD or burger, whatever, these foods when they get processed inside our body, the trans and all, they results in the formation of ROS, reactive oxygen species. ये क्या करते हैं? ये हमारे अच्छे enzymes को denature कर देते हैं. And that's how we start getting, all the different kinds of problems. ऐसा नहीं है that we cannot replace, we can, but everyone has got a limit of having or synthesizing the enzymes. क्या पता वो particular enzyme मेरी body में mutated हो और बचा कुचा जो भी enzyme था खत्म हो गया. Now I'm not able to replace it. So such kind of problems will come. Okay. So that's why all maximum disease which is there in the countries is basically because of bad habit or a bad lifestyle. Okay, it's not because you got it from your parents, from the genetic and all. It's all because of bad lifestyle. Anyways, it's okay. I mean, even I do eat. I can't control sometimes, so it's okay. Yeah. So this is our Hayflick limit. So you can see number of cells. Time is there. So what you can see in the case of cancer cell, it is not going to reach its saturation. Right? Whereas in the case of the normal cells, it goes till around 50 to 70 generations. Okay, and after that it reaches its senescence. यानी कि जहाँ पे वो stop हो जाएगा divide होना, and eventually it is going to die. It will undergo apoptosis. Okay, so कोई भी cell जब मैं बोलती हूँ that it is eventually going to die and all, it means it is going the process of apoptosis. Okay. Now here one more thing I would like to ask. What is the difference between PCD1 and PCD2? Do you know, guys, what is PCD? It is another name of apoptosis. Ab bolo. Yes. So, what else do we call apoptosis?
Program cell death.
Yes, very good. Now, if I say that it is program cell death 1 and 2, which one is apoptosis over here and what is the other one? कभी सुना है?
No, ma'am.
No? Okay. So program cell death 1 is your apoptosis. ठीक है? जब हम cell signaling पढ़ेंगे, there we are going to discuss all this. PCD1 is your apoptosis, make a point of it, and PCD2 is your program cell death 2. This is different kind of cell death, which is also known as autophagy. Autophagy, I'm sure you must have studied. You must have been knowing also. है ना?
Yes, ma'am.
Yeah,
Yes,
but yeah, so when we do research, or if you follow good reference book, then you will see that instead of apoptosis and autophagy, many times they use word PCD1 and PCD2. Okay, okay, good. Okay, let's do the MCQs, and with this, the DNA replication is over. Okay, so maybe from Tuesday or Wednesday, okay, we'll start with the new topic. 3 more topics are left, that is DNA repair, transcription, and translation, and of course their regulation. So with that, we'll complete our mole bio. So maybe by Tuesday or Wednesday we'll start. Monday is hectic for me, so that's why I'm not taking on Monday. Otherwise, Tuesday or Wednesday we'll start. Okay. Yeah. So let's do these MCQs. So your first question is, telomerase activity was monitored in the following cell types. The highest amount of telomerase activity was found in the combination of dash. So yes, guys, what do you think? What will be the answer?
A.
ma'am. A maybe.
A.
Everyone is saying A. Tanvi, what is your answer?
Yes, ma'am. I said A.
Okay, okay, yeah, okay. So the correct answer is A, embryonic and hematopoietic stem cells. Good. Second, telomerase. So there are 4 sentences. A telomerase is a ribonucleoprotein. It participates in replication of linear standard DNA. It is species specific. It is absent in somatic cells. So which statements are correct? Yes.
I think it's A.
A.
PQ and
A,
R.
ma'am. PQ.
Everyone is saying A.
Yes, ma'am.
The answer is C. Absent in somatic cell, which is also true, right? You are okay
Yeah.
with PQR, but it is also true, right? That telomerase is absent in
Yeah.
somatic cells. So the option is C over here. All or all the above, whatever. Okay. Okay, here this is also very important thing. So I made the diagram, made the table over here. So these are the names of the proteins, okay, and these are the names of the antibiotics, okay, which might be asked in the exam, like what kind of antibiotic or inhibitor, is able to inhibit these particular enzymes. Okay. So for example, in the case of bacterial gyrase, it is novobiosine nalidixic acid. In the case of mammalian topoisomerase, okay, so it can be 1 or 2 mammalian. It is etoposide. Okay, it is like this. It is etoposide and adriamycin. Then DNA breakage and rejoining process, ciprofloxacin, quinolones. So you have got quinolones and derivatives. तो थोड़ा नाम में भी ध्यान देना, जहां पे भी क्यूनोलोन्स फ्लूरोनोन्स आ रहा होगा तो समझ जाना, they are asking for this DNA breakage and rejoining process. Now suppose it is binding to the DNA through intercalation. Intercalation मतलब क्या होता है? DNA के बीच में जाके बैठ जाना, like how your ETBR goes in binds, right? So the inhibitor is going to be actinomycin D. Now cross-linking the strands, mitomycin. Clear? Now, based on this, there was one more question from previous years. So, which inhibitor can inhibit the action of gyrase? Okay, you must not be able to remember, but answer क्या होगा हमारा फिर यहाँ पे ऑप्शन में क्या क्या दिया हुआ है? So we know the inhibitor of bacterial gyrase क्या है? Novobiosine and nalidixic acid. ठीक है? अब आंसर बताओ.
Nalidixic acid.
Yes.
B.
Okay, so answer is B. So please remember that table. Okay, I think this is the last question. Okay, so telomeres are the repetitive sequence of T and G, which helps in cleavage from the nucleases. So these telomeres are synthesized by the enzyme telomerase. Which of the following are the properties of enzyme telomerase?
D.
Okay, one person said D. What about the others?
E, ma'am. All of the above.
So it is a reverse transcriptase. Yes, it consists of RNA template. Yes, it translocates 3' end of the DNA to synthesize repeated sequence. Yes, correct. So the answer is all of the above. Good. Okay.